Good morning, everyone. Welcome to the 10th Annual Alzheimer's Drug Discovery Foundation's Fall Symposium. We're going to have the panel shortly. My name is Mark Roithmer. I am the CEO with ADDF. I'm delighted to welcome you today. It's been a very, very exciting 12 months since we were all together last year. Let you know, 10 years ago, we created Hope on the Horizon, our annual fall luncheon and symposium, which honors philanthropists who make a significant contribution to Alzheimer's research. It is an opportunity to thank our donors for their generous support and share with you the progress, and I say progress, that we are making to getting closer to finding new treatments and cures. The last 2 years alone, we've seen exponential growth from leading philanthropists, including Bill Gates and Jeff and Mackenzie Bezos, among others, for our Diagnostics Accelerator program, a new initiative that shows promise in developing better diagnostics tools. This year, we awarded funding for the first round of recipients earlier in the year, and just a week or so ago, we funded our second round of recipients. With the right tools in place, we can deliver better designed clinical trials and get treatments to patients faster. You will hear this morning from Dr. Howard Fillett and our esteemed expert panel about ADDF's investments in new and diverse targets to treat Alzheimer's and help drug programs advance in clinical trials. And we are thankful to all of you for your continued support of science, and we are particularly grateful to Lily, Mary Beth Howlett, and Greg Johns, who are here today. They support our symposium year in and year out, and if we could have a round of applause for Lily, please. Working with all of you, we can reach our shared goal to accelerate the development of drugs for the prevention and treatment of Alzheimer's. 21 years ago, this organization was founded by Ronald and Leonard Lauder, driven by their passion and perseverance to identify a cure for Alzheimer's disease. It is my privilege to bring to you our co-chair, co-founder, Mr. Ronald Lauder.
Good morning. I must tell you, more and more I'm recognizing many, many people year in, year out, and I thank you. You're about to have a fantastic program today. And thank you, Mark, for your gracious introduction. Well, I was watching, made sure you didn't miss anything. It's my pleasure to welcome you to our 10th annual Fall Symposium. When my brother Leonard and I started ADDF 21 years ago, back in 1998, there was almost no hope for Alzheimer's patients. If someone you loved was diagnosed with Alzheimer's, it was worse than a death sentence. It was, and still can be, a long and painful goodbye. But Leonard and I would not and could not believe that nothing could be done. So we contacted Howard Fillett, in the front row here. Howard, you all know him. We told him what we wanted. We threatened him if he didn't get it. And now, 2 decades later, he's aged 40 years, and his work has been fantastic. And the people at ADDF, and all of you who have supported this effort, that long and painful death sentence is beginning to change. There is hope on the horizon, and we are becoming more hopeful with every passing year. Let me repeat the last line, because all of you in this room need to hear it more and more. There is hope on the horizon, and we're becoming more and more hopeful every passing year. That optimism comes from the work of ADDF and our willingness to take big risks. In particular, our focus on novel and repurposed drug programs. Repurposing is taking an already approved drug to treat another disease or medical condition, and it means accelerating treatment for Alzheimer's disease. In today's symposium, Hope on the Horizon, Repurposing Drugs for Alzheimer's disease, you are going to learn more about some of these drugs in the pipeline, which are as diverse today than ever before. Our recent clinical trials reports showed that there are around 100 drugs in clinical trials, with the ADF providing support for 20% of these trials. That's 20 trials out of 100. There's no one else doing it. 2/3 of ADF support trials are in phase 2. That's the phase when drugs are tested for their effectiveness. For the first time, and one even entered phase 3, the final step before FDA approval. Nearly 60% of ADF trials are focused on prevention, primarily to prevent the progression of mild cognitive impairment to dementia. We'll hear a great deal about it today. Several classes of repurposed drugs are in their clinical trials. The development of these repurposed drugs, an area of research that the ADF has always funded and promoted a fast-track effective treatment for Alzheimer's. Len and I created this foundation with the goal of finding the best ideas for new drugs and then fast-track them to clinical trials. There are 3 panelists today. Each had a great idea of taking an existing drug on the market and repurposing for Alzheimer's patients. The ADF provided them with the financial support to explore their ideas through clinical trials. We are thrilled to have them share their work with us today. We are changing how we treat Alzheimer's, and we can never have come to this point without your support. And our Chief Scientific Officer, Howard Phillip. Yeah, nice to you, Howard. We'll be leading. Howard will be leading the panel discussion in a few minutes following the symposium. We invite you all of you to join us for lunch. This year, we are honoring Pamela J. Neumann, President and CEO of PGN Strategies, for a tremendous commitment and tireless effort on behalf of our mission. Thank you, and please enjoy the panel discussion. I must tell you, I'm going to the back of the room because I have to call Israel a couple of times. And they have this thing on Friday, you can't call too late. If I go back and forth, I decided I didn't want to run the back. Anyway, today's discussion, I've spoken to the people who are involved. It's fascinating. And this could never have happened 5 years ago. Am I right? And ADF, I must tell you, if we do anything, this was one of the greatest things we're doing in the prevention of Alzheimer's. With that, I leave you for a fantastic discussion. Thank you all for being here. My brother Leonard would be here, but he had a back operation, and for some reason he couldn't get here enough. We were going to carry him in, but he felt that was the wrong thing to do. Yeah. Anyway, thank you all, and have a fantastic discussion.
Thank you, Ronald. Before Dr. Phillip welcomes the panelists, I would like to introduce to you a representative for a very special presentation. Barbara Goodman is the president and CEO of Cures Within Reach, who will be presenting their 2019 Global Health Repurposing Awards Patient Impact Philanthropy Award to the Alzheimer's Drug Discovery Foundation. A big thank you to Barbara, Claire, and Cures Within Reach for joining us today, and we appreciate your continued support, and come on up.
I'll put this right there. Good morning, everyone. Thank you very much, Mark. My name is Barbara Goodman, and I'm the president of Cures Within Reach. We're a Chicago-based not-for-profit that raises funds and finds repurposing treatments and helps bring them to commercialization. We help bring them to follow-on funding, and we catalyze the opportunity, not only in neuro and neurodegenerative diseases, but we also focus on rare diseases, we focus on oncology, we focus on many, many other opportunities. And this year, we selected ADDF to receive our 2019 Patient Impact Philanthropy Award. The reasons we chose ADDF could not have been more appropriately summarized by Mr. Lauder himself. ADDF's leadership in bringing repurposing to the forefront of opportunities for faster patient impact and faster treatment. We are thrilled to honor ADDF this year with this award, and we're thrilled to have one of our co-funded researchers, Dr. Krista Langto from the Sunnybrook Research Institute in Toronto, as a great example of the collaboration opportunities between not-for-profits like Cures Within Reach and ADDF to fund research that quickly brings follow-on funding, next stage of research, publications, and clear path to patient impact. And in the case of Alzheimer's, caregiver impact as well, which is critical. Past awardees of this award include large not-for-profit disease groups like Leukemia and Lymphoma Society, as well as philanthropic organizations like the Robert Wood Johnson Foundation. I'd like to officially bring Dr. Howard Phillott up to receive this award from Cures Within Reach.
Thank you. Welcome, everyone. Thank you so much to Barbara Goodman and Claire Thibodeau of Cures Within Reach for giving us this wonderful award. I have to say it means a lot to me personally. We have been supporting repurposing, and we've been champions of repurposing for many years now. And to be recognized for our efforts in this is very personally rewarding. I really appreciate it. As Ronald mentioned and Mark mentioned today, you're going to hear from 3 scientists who are involved in repurposing. Currently, we have 16 repurposing trials going on, 16 different drugs that are currently on the market, that are currently approved for other diseases, that we're looking at the mechanisms. Our scientists are constantly looking for these kinds of opportunities, and then working with investigators to tailor clinical trials to test these drugs in Alzheimer's disease patients. And of course, the advantage of that is that where it takes 12 to 15 years and $1.5 to $3 billion to bring a new drug to market, with repurposing, we reduce the cost by more than half, and the time frame to getting the drug to patients can be as little as 3 to 5 years. Once a repurposing trial like you're going to hear about today has positive results, it's possible for doctors like me to prescribe those drugs the next day. And I can tell you that off-label, based on the science that we've seen, I'm already doing that for some patients. So we're going to hear, though, about the science today of what this involves. Recently, you've heard about the Biogen study, and I'm sure many of you have questions about that. Obviously, that's an anti-amyloid strategy. I'm happy to discuss that, but not right now. I don't want to say that what that anti-amyloid strategy means right now, other than to say that we've had a lot of failures. The recent results are kind of interesting. We're not sure where it's going to go yet. Kind of the angel is in the details with the Biogen study. Our scientific strategy for almost 10 years now, for more than 10 years now, has been to go beyond amyloid. We did fund a few amyloid programs in the early days of the foundation when there were no drugs in the clinic for anti-amyloid strategies. In fact, ADDF, through the Oklahoma Medical Research Foundation, spun out a company at the time called Zapac, which brought the first anti-amyloid pill into patients in clinical trials back more than 10 years ago. So we were interested. When Big Pharma came in with billions of dollars, it was time for the foundation to move on, basically, and let Big Pharma and all the things that they can do in an excellent way to really test the hypothesis that amyloid does or doesn't cause Alzheimer's, or more importantly, whether amyloid is a rational therapeutic target that can help people. Our strategy is recognizing that the leading risk factor for Alzheimer's disease, far and away, is aging. And in fact, Alzheimer's, if you think of it, is much like cancer, which is a disease of aging. It's much like heart disease, which is a disease of aging. It's much like chronic renal failure and hypertension and diabetes. These are all diseases of aging. And what that means is that if we learn from the biology of aging, and I'm a gerontologist by scientific training as well as a neuroscientist, it means that all these diseases are caused by multiple pathways that are related to aging, oxidation, inflammation, something called epigenetics, vascular problems with low oxygen to the tissues. And in some ways, the brain is just another end organ for the attack of all these mechanisms of aging. And so what we've done is we've built a portfolio that's based on the biology of aging, where the largest component, for example, of our therapeutic portfolio is inflammation. It's been shown recently that people can have amyloid in their brain, but if they don't have inflammation, they don't get worse. And so we're supporting a number of anti-inflammatories for Alzheimer's disease. As I mentioned, we have a long history of supporting repurposing drugs. Right now, we're supporting 16 clinical trials for repurposing of drugs. Let me give you some examples that you probably have heard of that kind of relate to success in this field. Cyclosporin was originally developed as a drug for cancer. It was then repurposed for organ rejection and also psoriasis. Raloxifene, or tamoxifen, was originally developed for osteoporosis, and now it's approved for the prevention of breast cancer. Aspirin was originally used as an anti-inflammatory and a drug for pain, and is now used in heart disease patients as what we call an antiplatelet agent. So there's many examples of repurposed drugs that have gotten to market. And one of our challenges is to figure out how the positive results that you're going to hear about today. And I should emphasize that. We're going to report to you on positive results. You've heard so much negativity. You're going to hear about positive results from these clinical trials today. Why is philanthropy very unique in this niche? Because of some of the commercial issues in repurposing, nonprofits can support repurposed trials where big pharma and venture capital investors, for example, won't do that. And there's various reasons for that that are structural in the commercialization of these drugs that I won't talk about today, but that's something that the ADDF wants to address. So with that brief introduction, I'd like to introduce our first panelist, Krista Langto. Krista, we've worked with for actually many years in different spheres. Krista is the senior scientist in geriatric psychiatry and head of the Neuropsychopharmacology Research Center in the Hurwitz Brain Sciences Program at Sunnybrook Research Institute and also affiliated with the University of Toronto. And she's published more than 300 papers and came to us with an idea about using cannabinoids, which is something you hear about a lot these days, for Alzheimer's disease. And let me just, if I may, kind of frame as a clinician what the problem is here. First of all, the main problem is, and for those of you who have cared for loved ones with Alzheimer's disease, you've seen it up close and personal, how bad agitation can be, how disabling agitation can be, not just for the patient, but for the caregiver. Agitation is one of the leading reasons why Alzheimer patients end up in nursing homes, and it's a leading cause of morbidity in caregivers. And there are no drugs approved by the FDA for the treatment of agitation in people with Alzheimer's disease. And that's changing now. There are companies out there that are developing drugs for agitation. The need's been recognized. Krista had a terrific insight based on what are called CB1 and CB2 receptors. There's a drug on the market, which I'll let her talk about, and I don't want to steal your thunder, but just to say that there's a drug on the market in both the US and in Canada called Nabilone that she's going to talk to you about. It's a synthetic THC analog. And one of the more interesting things to me about it, which we incorporated into the trial, is that it's not only going to be able, and she'll show you the data, to be safe and effective for agitation, but in addition, it may have anti-inflammatory properties that might be useful in the treatment of patients with Alzheimer's to slow the course of their disease. So with that, let me introduce Krista.
Thank you very much, and thank you for that introduction. And you did cover a lot of the things that I was going to cover. But first, let me tell you a little bit more about the drug I tested. I tested Nabilone, and Nabilone is actually approved for nausea and vomiting associated with chemotherapy. So why would you take a drug like that and use it in Alzheimer's disease? And Dr. Phillott already told us a little bit about that. I looked at that because it actually works through the endocannabinoid system. So Nabilone is a synthetic version, so a lab-made version of THC, or tetrahydrocannabinol, and we all know that that's one of the major ingredients in marijuana. So it has a lot of other effects that actually have not been taken advantage of. So we wanted to target the endocannabinoid system. When you give something related to THC, there were 4 clinical things that we were very interested in. Clinically, you're going to see that it has a calming effect. And that's dose related. So you can make someone really sedated, but we were looking for the sweet spot that would maybe help with agitation, so just calm people. And it does other things clinically that we thought might be useful. It actually, because it's approved for nausea and vomiting, we know that it can increase appetite, right? We've all heard about the munchies. So why would that be important in Alzheimer's disease? When people are agitated, they're frequently too agitated to sit down and eat and take regular meals, and that really decreases their quality of life, makes them weak, and makes them prone to falls. So people, as they have Alzheimer's disease, the more severe it gets, they tend to have weight loss. So we wanted to maybe help with that appetite. It has a third effect that might be potentially beneficial. We know that it's also approved in some other diseases for pain. It can help with pain. And we know that untreated pain can also contribute to agitation. Finally, it can help with anxiety. And we know that sometimes people, especially even starting in the mild stages of the disease, but in the areas that we were targeting, in moderate to severe Alzheimer's disease, can have a lot of anxiety. There's a flip side in that it can also contribute a little bit to memory impairment, depending on the dose. So that's why, in particular, we thought people with moderate to severe Alzheimer's disease, if we could get the dose right, we wouldn't see very much memory impairment that would interfere with their quality of life. But for all those clinical reasons, we might see an improvement in their quality of life. And the second major region is what Dr. Phil had already told us about, right? The medications that are used for agitation right now have a black box warning on them that says that they can increase your risk of mortality. So we know that they're not protective of the brain, but by stimulating the cannabinoid system for a calming effect, we may have that. And there's 2 reasons. You heard about CB receptor 1. So the cannabinoid receptor 1 is responsible for something that's called excitotoxicity. And that's the process by which neurons are killed by overactivation. And we know that in advanced Alzheimer's disease, that's going on. Well, it turns out that in animal models, if you give this particular drug, it helps with this excitotoxicity. So it may have a protective effect at the brain for something that's going on late in the disease. And the other reason that you already heard about is neuroinflammation, so inflammation that's going on in the brain. Through cannabinoid receptor 2, it can actually help with neuroinflammation. So we wanted to try this medication for agitation. Dr. Phil already did an excellent job of telling you the problems that we have with agitation. There are currently no treatments on the market. It decreases quality of life for the patient for sure. The number 1 diagnostic criterion for agitation is that the patient is in distress. So we hate to see our loved one in distress. They can have verbal agitation, yelling and screaming and threatening. They can have physical agitation, where they're physically aggressive against caregivers and the people that they love, even though that's not the personality of that person that you've seen their whole life. So it's very disturbing to the patient themselves, to the extent that they have insight in their caregivers around them. And then you can have non-physical agitation. So not just calm, happy wandering, but an agitated, distressed physical aggression. So that's agitation, and it turns out that by the time you reach the long-term care facility, between 20 and 50% of people will suffer from agitation at some point in the disease. So it's a big problem. So from that, we ran our trial. So I'd like to just stand here and thank again publicly ADDF, as well as Cures Within Reach, for coming up with this to fund our trial. So our trial looked at agitation in Alzheimer's disease, and we gave nabilone and placebo, and we were smart about it. We did a placebo run-in to make sure that people were really agitated, because you can have a big placebo effect. And we found, and then we gave each person both nabilone and placebo, so they acted as their own control. So I'm pleased to report today that our results were positive. We found a big effect on agitation, and in fact, the effect that we had on agitation was bigger with respect to effect size than any drug that has currently on the market that's being used for this. It's the biggest effect size that we've seen. The flip side of that is that we had to titrate it up, and we found that we did have sedation, but we managed to find the sweet spot where sedation wasn't interfering with the patient's everyday activities of living. So not only was it positive on agitation, but we took blood samples and we had some interesting effects on biomarkers. We think that before you give someone a medication, you should have a biomarker that gives you some hint of whether they're going to respond to the medication or not. And we found biomarkers that indicated that it was having a beneficial effect on this inflammation that we see. So this was a phase 2 trial, and you heard about a phase 2 trial. There's more work to be done. Before we change therapy with a medication, we want to do a phase 3 trial, so a larger trial in more sites. And that's the evidence that clinicians will look for before they want to change therapy. So I'm also pleased to tell you that with the help of ADDF, we are planning on running that big phase 3 trial, and we hope to have our first patients in Q2 2020. So thank you very much.
Thank you very much, Krista. Could you talk about your ideas about, real briefly, about how you would think of how nabilone would get to patients, given even right now, for example, the very positive results you've had in your practice? Would you be prescribing nabilone for patients off-label, for example, that were agitated?
People are prescribing nabilone off-label. We think that there's more work to be done. We have to find the exact dose still. We have to, because it was a phase 2 trial, we only looked for a very short period of time, and not everyone responds. Even though we had a big effect, not everyone responds. So there's more work to do to find out who's responding and who should be given nabilone. We interestingly also found that doing additional analyses that weren't part of the trial, that we had people who had higher levels of pain tended to respond with agitation. So we may want to even look at people who are having untreated pain and agitation associated with that, and look at it for a non-opioid method of pain control in this population. And also for people who are having nutritional difficulties. We only looked at that as a sub-analysis, and we may find that nutritional stuff is important. So there's lots of other things that it can be used for, and we want to make sure that it's healthy for the brain.
Thank you. We'll get to some audience questions at the end after the 3 speakers. The next speaker is Dawn Matthews. We funded Dawn when we helped her to create a first company called Abient, and now working as the CEO of ADDM Diagnostics. And the idea around it was that we can send people for MRIs of the brain to evaluate whether there's atrophy in the brain, whether the brain is shrinking according to Alzheimer's disease. But we can't quantitate it. It's always been very difficult to quantitate that. So in clinical practice, and also importantly in clinical trials, we want to be able to quantitate the volumes of the various parts of the brain that are infected by Alzheimer's on MRI, and make that information clinically useful in clinical trials. We also have other kinds of brain imaging, like PET imaging for the amyloid that our foundation helped to bring to market, the PET amyloid diagnostic, and PET glucose utilization, which allows us to look at the function of the brain. And so so Dawn developed this wonderful technology, and then we introduced her to Jeff Cummings, and she's run a clinical trial where Dawn has turned out to be the lead, actually, on the report of how rasagiline, a drug for Parkinson's disease, we repurposed for Alzheimer's, found positive effects. But I think one of the critical things in demonstrating those positive effects was Dawn's technology and enabling us to analyze the imaging that was done in the trial. So let me introduce Dawn. And I think I'm going to ask, Dawn, if you don't mind, if I could ask you to sit. Thank you. You're good.
Thank you so much. It's really a pleasure to be here, and I really want to thank Dr. Phillip and the ADDF, the Louder family, for the funds that have enabled these trials. Just as a comment, I was able to participate in the September conference that the ADDF held, and it really was the most heartening, encouraging conference I have attended, and I've attended many of them, both by the positive results that were being conveyed, as well as the spectrum of different, I think I'm getting some feedback, the spectrum of different approaches that are being tried. So here I'd like to talk to you about the rasagiline trial that, as Dr. Phillip mentioned, was led by Dr. Cummings from the Cleveland Clinic, and we were fortunate to participate in leading the imaging. So rasagiline is a drug that is currently approved for use in Parkinson's disease. You might have heard of it as Azilect. It was originally approved for helping with motor function, but in studies of Parkinson's patients, they noticed that it also had an effect, a positive effect on cognition. A close cousin of rasagiline was also found to have a favorable effect potentially in Alzheimer's patients, but that drug has some side effects that are not positive. So with the study of rasagiline, it was designed to test whether this drug could have a positive effect on a variety of different aspects of cognitive function in mild to moderate Alzheimer's patients. And that's a pretty broad spectrum of people that are pretty advanced in their symptoms, some of them milder, but it was a really important study because that's an underserved part of the Alzheimer's community. And so this was designed as a proof of concept trial. It was a phase 2 trial, but what I mean by proof of concept was that it was designed to efficiently determine, did this drug have enough promise to merit a larger, more costly trial to power it up? And so this trial involved 50 Alzheimer's patients. 25 of them were put on placebo and 25 on the drug rasagiline. And this was done for a period of 6 months, so it was a rather short study. And in order, typically in a study, it would take many more patients to determine whether there was a beneficial cognitive effect, because cognitive tests can be subjective, and if you don't know who you really have in your population, you have a lot of variability. So there were some key aspects of the study designed to make it efficient. One of those was using FDG PET imaging, which Dr. Phillip mentioned, to select patients for inclusion in the study. Now, FDG PET measures glucose metabolism in the brain. Glucose is a primary source of your brain's energy, and it turns out that when people have Alzheimer's disease, the brain declines in its use of glucose because the neurons are getting sick and dying in a particular pattern that we can recognize and quantify. So we used our technology to determine whether patients had this pattern of glucose metabolism in their brain for inclusion in the trial. A second use of the imaging biomarkers was to detect brain changes during those 6 months or after 6 months of treatment. So again, we used FDG PET to detect whether there were changes in the brain's use of glucose. So that would be the health of these neurons in relevant places, relevant regions of the brain, comparing drug versus placebo. We also used the imaging to help identify how severe really were patients when we started the trial. Because one of the problems with these trials has been that not only have the wrong patients been included that don't even have Alzheimer's, but often you'll see people who aren't that severe, even though clinically they might seem severe, their brain is really not that advanced in the disease compared to people who are worse off. And that can really skew the results of the trial and interfere in finding something that might actually work. So one thing that we did too was we compared patients on the basis of severity. Finally, this trial was a first of its kind in using tau PET imaging as well. So in addition to measuring glucose metabolism, we were able to use another kind of imaging that measures an abnormal protein called tau. You may have heard of amyloid and tau. Tau in the brain. And that was really an amazing feat, I think, by Dr. Phillott and Dr. Cummings to get this kind of imaging in the trial. So the results were very exciting. It was a pleasure to be in a trial that had a positive outcome. It met its primary endpoint, which was to determine whether patients who were treated with rasagiline would differ in their brain changes as measured by glucose metabolism versus patients who were treated on placebo. In addition, although it wasn't necessarily powered to detect clinical endpoints like memory and thinking, executive function, quality of life, they did measure approximately 8 of those kinds of parameters. And lo and behold, there was a directional effect of patients treated with rasagiline doing better in all regards and statistically better in both quality of life and executive function than the patients who were treated as placebo. So this data really gave, I think, impetus or a foundation for concluding that it would make sense to study this in a larger trial. So that's what Dr. Cummings is working on now, is securing the funding for a larger trial of rasagiline, using as evidence the imaging data and then the fact that the clinical data was consistent both with the imaging data as well as with the prior findings in some of these studies. So a talk about a study that included imaging wouldn't be complete without showing you at least a picture or two. So if I could have a slide, if you could back up, please, to the very first slide in that set. There we go. So what you're looking at on the left, there are the images. The gray images are pictures of the brain taken with an MRI. So that's showing you brain structure of a template brain from 3 different slices, this way, sideways, and front on. So what you're looking at there in the orange and red are regions of the brain where the function of your the neurons of people with Alzheimer's disease declines over time, and so does the use of glucose. So we found that the placebo-treated patients in the graph on the left all progressed in their quantitative score of that pattern. We saw some benefit in the rasagiline patients in that regard. However, where we really saw the most difference was in the frontal and striatal regions shown on the right. Those would be parts of your brain that are involved in the executive function and quality of life that were improved or did not decline in those patients as much as placebo. The bottom is a graph in our paper to be published that shows that there was a significant difference in the brain's function in those regions. The next slide just shows you with 30 seconds or 20 to go. If you can just go to the next slide. There. So on the left at the top is a patient with Alzheimer's disease, and the dark blue areas show you where their function has declined. We were able to learn from this study not only that there's a large variety of people, large variation in both FTG and tau, but that the tau pattern, which is on the bottom, as well as the MRI pattern, which is shown in our report on the right, were very closely related to what we were seeing with glucose metabolism. So it was a very rich and valuable study for understanding how we might use all of these markers to plan studies in the future and to better understand the patients so that we can treat them better. So with that, I've used my time, and yeah, thank you so much.
So let me just, yeah. Let me just point out, Dawn's very humble, and I want to thank Avid Radiopharmaceuticals and Lilly for making the tau imaging agent available to this study, which was
Definitely.
That was a big deal. But tau imaging, so the tau represents the dying neurons. So what Dawn is telling you is that while the amyloid imaging tells you about the amyloid deposits, the tau imaging actually tells you about the dying neurons. And now we can quantitate that. And we know that the tau imaging correlates with cognition. So we have what's called a surrogate biomarker. In future studies, we can measure tau imaging and expect that if tau imaging improves and there's less dying neurons, that will translate into a clinical benefit. And this study illustrates how we're learning how to do clinical trials in Alzheimer's disease better. We're making the trials more rigorous, more quantitative, more objective. We're enrolling the right patients. And I think this is going to be a landmark study in the field. It's not a big study. It didn't cost $10 billion. But it's the first study to use tau imaging in a clinical trial and show a benefit of a drug on tau imaging. And so it's a really important study. Thank you so much, Dawn. Our last presenter is Anna Pereira, who is a cognitive neurologist and neuroscience at the Icahn School of Medicine at Mount Sinai and also at Rockefeller University here in New York. And she's going to tell us about a neuroprotective agent that is called riluzole, again, a repurposed drug originally brought to market for the treatment of ALS or Lou Gehrig's disease. The only drug on the market for Lou Gehrig's disease that's ever been shown to slow the course of the disease. And some of the mechanisms by which riluzole works are directly applicable to the treatment of Alzheimer's disease and the slowing down. We're very excited. Anna's still in the process. She's been amazingly persistent in getting this trial done. It was a really hard startup at Rockefeller, and ADDF has stuck with her for I don't know how many years now, but I know we're enrolled and we're going to get results in a few months, and I'm anxiously awaiting the results because at least I think it's a very exciting drug to potentially be used in Alzheimer's disease.
Thanks, Howard. Yeah. Maybe I'll talk a little bit about my trajectory and that led to the program that Howard has mentioned. Actually, when I was in medical school, I translated several chapters of the Principles of Neuroscience, which is the major textbook in neuroscience, edited by Eric Kandel, the Nobel Prize, and many others. And at that time, I really realized how complex, the most complex machine is really the brain, and how fascinating it is. And that I wanted to understand the biology and study the biology of those cognitive processes, how memory, language, emotion, how those processes occur in the brain. So I came to Columbia University to train as a neuroscientist, and then to complete my medical training, I completed my neurology residency at Harvard, and subspecialized in cognitive neurology since I'm interested in cognition. And at that time, I was committed to become a physician scientist. And the major goal for physician scientists is really to make discoveries that you can have potentially an impact in human health. And at that time, I decided to focus my research on Alzheimer's disease because it's the most common neurodegenerative disorder. And as all of you know, it's patients with Alzheimer's disease is slowly and progressively start with memory loss, but affect multiple cognitive functions. And all of those key abilities, they make us who we are. They give us our identity. So it's a devastating disorder. So I was at Rockefeller for many years, and more recently at Mount Sinai, where I see patients with at the Center for Cognitive Health with cognitive syndromes, including Alzheimer's. And I lead a laboratory to understand the neurobiology of aging and Alzheimer's disease, really in order to develop novel targets and novel treatments. So my lab, more specifically, has been trying to understand why neurons, they use glutamate, which is a neurotransmitter chemical through which neurons talk in the brain, why those neurons are more susceptible to having synaptic loss as you age, as well as neuronal loss with Alzheimer's disease. And those neurons are also the ones that mostly accumulate tau pathology and those tangles that Howard was talking about, which are very toxic and lead to neuronal loss. And actually, the best predictor of cognitive decline are really how those synapses, glutamatergic synapses and glutamatergic neurons work. So that's why you're targeting those neurons and those synapses in my research. In my lab, more recently, we're also trying to understand how intermittent hypoxia, which is low oxygenation that occurs in patients who have sleep apnea, why they have an increased risk and progression to develop Alzheimer's disease. So the biology of the glutamatergic system as well a little bit more. Not only are those neurons more susceptible to die in Alzheimer's disease, but you probably have heard about amyloid beta and tau many times. The release of these proteins and the propagation of tau from one area of the brain to the other is really dependent on glutamatergic activity. And I think there's a very growing literature and evidence showing that it's really when you accumulate tau as you age in an area of the brain called entorhinal cortex, and when that propagates to the cortex of the brain, that's when you really develop dementia. So my lab has identified and others that there's a major glutamate transporter in the brain, actually just published a couple weeks ago in PNAS, that if you downregulate this glutamate transporter in transgenic mouse models, it can accelerate age-related cognitive decline, and it overlaps molecularly with aging and Alzheimer's disease. So I have been working, as Howard pointed out, for many years on this medication called riluzole, which is currently FDA approved for the treatment of another neurodegenerative disorder, which is ALS. We have published in 2014 in PNAS that it could prevent age-related cognitive decline in mice and rats, and it produces neuroplastic changes in the brain, which strengthens neurocommunication. We later on published that it could recover many molecules, gene expression associated with aging and Alzheimer's disease. And more recently, we have also shown in animal models that it can affect microglia, which is very important immune cells and inflammatory cells that is involved in damage of neurons in Alzheimer's disease. So given the biology that we have developed, we wanted to translate those findings into patients. And this could not have been done without the support of the ADDF and Howard Phillip throughout the career. So we're very thankful to the Dana Foundation as well. So we're very thankful to all the ADDF team, Howard, and all of you who have been supporting Alzheimer's research. So the trial is a double-blind, placebo-controlled trial in which we use neuroimaging biomarkers, state-of-the-art neuroimaging biomarker, as Don has pointed out. We're doing FDG PET, which looks into metabolism of the brain, which is also a great predictor of how those neurons are functioning, in collaboration with Don, who's been amazingly collecting, analyzing those scans. And we're doing MR spectroscopy, MRI, and as well as neuropsychological testing, so battery of testing to objectively quantify your cognition. So this trial will be completing it soon, which will, and I think it's very important to emphasize that how important it is for us to really continue to try to understand the biology of Alzheimer's disease. And I think my lab and many others in my lab have been trying to really use a lot of modern molecular biology, as well as such as RNA sequencing, as well as structural biology tools, so that you can really try to identify using several models, animal models, postmortem Alzheimer's brain tissue, and in patients through neuroimaging, trying to identify the key drivers of Alzheimer's disease so that you can more effectively target the disease and come out with effective treatments for this disorder, for this devastating disorder. Thank you so much.
So Anna, you came out of the lab and decided to do a clinical trial one day, about 4 or 5 years ago, maybe, something like that. 5 years later, we're finally getting to the end of this. I don't think people realize how hard it is to do a clinical trial. I mean, why does it cost, big pharma to run a phase 3, $400 million, when half of the $400 million, $200 million is for enrollment, for finding the patients and getting them into the study? And I know you struggled tremendously at Rockefeller when you opened up the Alzheimer's clinic there, finally got a second site at Mount Sinai. Maybe you could tell us, how hard was it to get this trial going?
Yeah, well, Rockefeller is a unique place. It's a wonderful place. It's really basic science-driven, less of a medical center. But you know, it takes a lot to run a trial. Actually, it's much simpler for me to be in the lab. But to recruit patients, and I've got to say that patients who enroll in trials, they are partners of discovery. It's really, we could not advance the science without them. And they are really at the forefront of generating knowledge. So extremely grateful for all those patients who enroll in those trials. And it's very hard. There are many trials. Recruiting, getting those patients in the trials is hard. And it's, Alzheimer's is a very complex disorder. So the neurons, they're really responsible for higher cognitive functioning are getting damage. So the ones that form association will integrate all the information, because you can see the primary cortices are fine. People can walk, can feel, but they can't really integrate the high processing of information. So, and it takes a long time. So those trials are lengthy. They are those neuroimaging tools that are really important to measure in a quantitative and robust measure the structure of the brain, how much tau pathology occurs in the brain. Those are very expensive imaging tools. But I think it's very important that we don't give up the fight and really try to find an effective treatment for this disorder.
Thank you. I want to open up the panel to questions from the audience, if there are any questions. Yes, sir. Howard.
There's a question about blueberries as being a factor in diet. That blueberries would be a factor in diet. And I was wondering what your comments might be on that subject.
Well, Howard, you're opening up the issue of prevention here, I think, and also what we can learn from foods, for example. There have been a lot of prevention studies. There have been some prevention studies now, both different kinds of evidence, what we call epidemiological, observational evidence that can only prove associations. So there's been associations of Mediterranean diet, for example, with the prevention and slowing the onset of Alzheimer's disease. And then there's been some recent analyses that what's the key factor in the Mediterranean diet that might be associated with the slowing of cognitive decline. And it looks like it's berries, basically, berries and vegetables, and particularly the dark ones. And the way I look to what we think is that the dark berries, they live under the sun 24/7. So through hundreds of millions of years of living under the sun, Mother Nature is the most amazing chemist, and through evolution has developed incredible antioxidants that we can't synthesize and we can't really get from synthetic chemicals. There's never been a synthetic antioxidant that's ever been shown to have any benefit for any disease in the history of the world. But it looks like blueberries might work, and so it may be what are called the polyphenols in the blueberries. So we're interested to learn from that to say, okay, if it's the blueberries, if it's the antioxidants, how can we prove that? And how can we get polyphenols, for example, and run them? What we would want to do is run them into a clinical trial. And we do have one study like that, but these are very difficult clinical trials. But I think it's a good point about how we interpret data that we read about in the paper and actually try to get that into a therapeutic area. And we have a website on our website called cognitivevitality.org, cognitivevitality.org. And we have a report on berries and diets and polyphenols there if anybody wants to listen. Last year, and Dr. Hara is right there, who's chairing that in our aging prevention program. And last year we had over 500,000 visitors to our website for the Cognitive Vitality website, asking questions about various nutrients and supplements and diets and so on. And we have a deep analysis. We have 3 PhD scientists running that. So thank you for raising the issue of food. Appreciate it. I was going to, if there's any other questions. Yes, back there.
Knowing the importance of anti-inflammation, and since there are many available anti-inflammatory drugs, all of which have other side effects, but why don't are we working on anti-inflammatory drugs with fewer side effects that might also help?
Yeah, I mean, I can answer that. So there were studies showing that, for example, drugs like Motrin were, again, in what we call observational epidemiological studies, where you follow people over time and you look at their diets and everything that they do and whether they take Motrin or not, and then you look at the risk of Alzheimer's. And actually, the early studies in the 80s showed that these drugs like Motrin and ibuprofen were associated with like a 40 to 60% reduction in risk, but then it doesn't prove it. It only shows it only shows an association, right? So then there were clinical trials of Vioxx and Celebrex and Motrin for Alzheimer's, and they all failed. So we need new anti-inflammatories, and there's two ways that inflammation affects the brain. I mentioned how the amyloid and other proteins that get deposited can cause inflammation, and that really contributes to cognitive decline. That would be what we call neuroinflammation. But if anybody in the audience has ever been sick, you know that when you're sick, you don't feel well. And that's called sickness behavior in our research, and that's the effect of systemic inflammation on the brain. For example, in geriatric medicine, we teach medical students that if somebody gets pneumonia when they're young, the typical sign is cough and fever. But in an elderly person, the first sign of pneumonia in an elderly person is going to be cognitive impairment and delirium. And we're funding some studies right now looking at what's called postoperative cognitive decline. A lot of people after major surgery have cognitive decline, and we think that's from the effect of systemic inflammation on the brain. So there's two ways to address that. We're funding a biotech company that was spun out of Northwestern University with a drug called a P38 kinase, MAP kinase inhibitor. It's just a drug that targets this real important node in the biology of inflammation in the cells. And there's currently a phase 1 going into phase 2 clinical trial going on in Europe right now with that anti-neuroinflammatory drug. But we also funded at the University of Southampton a drug called Enbrel, which is one of the most widely selling drugs for rheumatoid arthritis. And we funded that repurposing study of that drug at the University of Southampton. Unfortunately, we need to repeat that study, but that's kind of some of the approaches that come to mind, both repurposing novel agents, reducing systemic inflammation as a way to prevent Alzheimer's, and then attacking the inflammation that's actually going on in the brain.
Also, more specifically as well, Howard, to adapt, there's a lot of genetic studies pointing out to microglia as a key mediator of...
These are the inflammatory cells, the microglia.
Right. What kind of inflammatory cells? There's a key immune cells in the pathogenesis, and now we're trying to understand the biology of it better so that you can target it the right way. This is a novel findings in the last few years.
We have a question.
As soon as I heard the term glucose metabolism, it made me wonder if there's any relation to type 2 diabetes, and if any of the drugs for diabetes might be repurposed for this cause.
That is an excellent question, and there is work going on right now. I don't know if you have any companies.
Well, we're funding a clinical trial of a diabetes drug at the Imperial College of London right now. It's a very large study being co-funded by the Medical Research Council of the UK, Novo Nordisk, General Electric. And we're really excited about it, and there's previous evidence, and I don't want to dominate the panel, but I would just say, just real briefly, and I'll turn it back to you, Dawn, that there's very good evidence now that diabetes is a risk factor for Alzheimer's. But you might want to talk about the glucose.
Right, there is a fairly large body of evidence that has emerged over the past several years on relationships between diabetes as a risk factor and also glucose utilization. Definitely a path to be explored. And then also the nutritional and lifestyle aspect. There's really a compelling body out there of evidence showing that it matters. So these are encouraging because it means there may be things we can do in our lives to impact the onset.
Yeah, and another repurposing study, metformin is one of the largest selling drugs in the world and the leading drug being used for diabetes. And we funded Jose Luxinger at Columbia University back several years ago to test metformin for Alzheimer's disease. And actually the primary outcome measure in that small trial was the FDG PET, the glucose utilization, to see how metformin affected. And it was a positive trial, actually.
It was positive.
It showed that patients with Alzheimer's disease who took metformin had better glucose utilization, better function in their brain. And unfortunately, Jose had a lot of trouble getting follow-on funding, but we just heard that he got, I don't know, $10, $15 million from the National Institute on Aging to run a phase 3 trial of metformin for Alzheimer's disease. Again, the leading drug for diabetes being repurposed for Alzheimer's disease.
And patients with Alzheimer's and diabetes, they progress much faster than those without diabetes as well. Another evidence for that.
Don?
Is a flu shot contain any elements which can affect Alzheimer's disease?
Anybody want to take that?
No, it doesn't. Short answer, no.
Yeah.
The flu shot is safe and it should not contribute to Alzheimer's disease. Just a comment, I think it's interesting so far that people are bringing up so many different mechanisms. And I'm excited because that's one thing that's been the strength of ADDF. And I think it's the way forward. We've focused on beta amyloid, but we've heard today that there are so many mechanisms that are in play, including neuroinflammation and, taking antioxidants. And I think, the field has moved. It's important that we have treatment, but we also have prevention. And part of this also overlaps with diabetes, but also the vascular effects. We're increasingly aware of the vascular contributions to Alzheimer's disease and how important it is to keep your brain blood vessels healthy. And we're also doing research in prevention where we look at those vascular effects and we're giving antioxidants and using lifestyle interventions. And I think the more different ways that we can attack Alzheimer's disease, the better chance we'll have of having a big impact. We're really going to need one of these multi-pronged approach. We have to start with looking at each individually, but I think a multi-pronged approach is what really gives us help for the future, and I commend you for looking at all the different mechanisms.
Thank you, and I want to reemphasize what Krista just said. If you think about cancer, most cancer patients are on 4 or 5 drugs. Most diabetics are on 2 or 3 drugs. Most hypertensives are on 2 or 3 drugs. And that's because I mentioned earlier the multiple mechanisms. So we're going to get to a world where we have better biomarkers, and we'll basically maybe 20, 30 years from now, because we are 50, 60 years behind cancer in research, the Imperial Cancer Trust Fund was established in the UK in 1902, and Alzheimer's didn't even describe Alzheimer's until 1906, and the National Institute on Aging that funds most research in academia and most basic research in Alzheimer's wasn't even established until 1976, and when I got into this in 1980, there was nothing known about Alzheimer's. So we're 50, 60 years behind cancer, but if you look at the cancer model, you have what's called precision medicine. You get tested, your cancer gets typed, and you get put on 3 or 4 or 5 drugs. And I think that's the future of our vision of Alzheimer's, but we got to chip away at it one at a time. And we'll ultimately have combination therapy. Don, I think you had a question right there.
I'd like, if possible, I'd like to go back to glucose again. I'm very much interested to hear you talking about how Alzheimer's affects the brain's ability to use glucose. My husband is in a memory care unit with Alzheimer's, and I have noticed one thing about, I would say virtually all of the people who are residents in that unit, they all crave sweets. Is that a side effect of, if that's because they're not purposing their glucose in the right way, or, and if it's true, is there more of a risk for diabetes because of that? I'd like to know.
Maybe I'll take that one. So the brain, the primary energy source of the brain is glucose. Okay, so that's the primary. So that's why these kinds of scans that Don does, FDG PET, it really measures uptake of glucose. And the neurons that are not functioning well, they uptake less glucose. So patients with Alzheimer's disease have a pattern in the brain of hypometabolism, less metabolism in specific areas of the brain, which is parietal, temporal, posterior cingulate frontal. To address your question about craving for sugar, so as the disease advances, it affects certain areas of the frontal lobe that is really regulating impulsivity and food, and that's more related to this sugar craving. It's when certain areas of the frontal lobes are affected rather than anything else.
Okay, we have a question here.
Would you comment, expand on
Sure. So the question is, would I comment on the Biogen results that we heard about last week? We've had many failures on this anti-amyloid, probably 20, 30 drugs that have failed on the anti-amyloid. The difference with the Biogen study, which is one of the things that's really interesting, is that it was I mentioned earlier how we're learning to do better clinical trials of Alzheimer's. And one of the reasons why Lilly bought the PET amyloid scan that ADDF helped to bring to market in 2012 through our funding at Penn back in 2000, from 2000 to 2004, is that the reason we funded that was we wanted to make clinical trials better and more efficient. And so at the time, Lilly was doing a clinical trial of a drug called selinezumab. And they'd used this brain scan to see if the people who were being enrolled in the trial actually had Alzheimer's disease, and they actually had amyloid in their brain, which was the target of the drug. And it turned out that 30% of the people who were enrolled in the trial by dementia experts around the world didn't have amyloid in their brain, and they didn't have the target of the drug, and they didn't have Alzheimer's disease. So of course you couldn't get a positive trial out of that. So now Biogen, so it could explain all the failures that we've had. Biogen comes along, and it's the first clinical trial where 100% it's required that you have a positive PET amyloid scan, the same technology that we developed, to get into the trial. And they used the drug as a monitoring biomarker, not just an inclusion biomarker, but as a monitoring biomarker to see if the drug was working. In other words, did it decrease the amount of amyloid in the brain of patients? And in fact, the drug worked. The drug decreased the amount of amyloid, so by 18 months, 80 to 90% of the people in their Biogen trials had complete absence of and complete removal of the amyloid from the brain. So now we have, for the first time, a rigorous scientific study, a clinical trial, that can actually ask the question, if you remove amyloid from the brain, what effect does it have on cognition? And here's where you get into how clinical trials are so complicated. Biogen's got a $3.2 billion program going on. They do a study called ENGAGE. It's 1,750 people where they get increasing doses of this drug to remove the amyloid from the brain, and it's completely negative. Completely negative. Then they do a second study, same design, same study, 1,750 people, called the EMERGE study, and it's very positive. How can that be? So part of the difference was that the people in the EMERGE study, who actually had ApoE4, they drove the study into people that had ApoE4 because they were most at risk. The people in the study that went on the highest dose actually were the responders. In other words, they were the people where the drug not only removed the amyloid from the brain, but they got better. The people had a slower rate of decline. And so it seemed to indicate in the EMERGE trial that the drug was clinically efficacious. And they also went back and looked at people in the ENGAGE trial who were titrated up to the highest dose, and they saw that those people also seemed to respond. The problem is from a regulatory, so the scientific experiment was really interesting, but the regulatory science here is going to be really difficult, because the regulatory challenge for FDA is, should we approve the drug? And they rarely approve the drug. If you're sitting at the FDA, right, and you've got this drug that literally is going to cost $50 to $100 billion a year potentially, and imagine that the entire Medicare pharmacy budget is about $450 to $500 billion. And this is a drug where elderly people, anybody with a memory problem, can come into the clinic and get a monthly infusion. It's a big burden on patients and expensive. And so the long and short is that I think from the problem here is the regulatory science. Would the FDA approve a drug like this where they have one negative study, one positive study, and some fancy dancing, let's say, the way that the data were analyzed was called into question, and I won't go into the details of that, but I think a lot of experts are calling into question, having concerns about how the data were being analyzed. So bottom line is, it's again, science is incremental. I think we have a glimpse that if we do the study right, the beta amyloid drugs may work, but I think they're going to have to do a third trial to prove to the FDA that this really is effective, and that's going to take some time. That's my opinion and the opinion of a lot of my friends. I think it just illustrates how hard and expensive this really is and how we're learning how to do clinical trials better.
Hi. We've heard a lot about the impact of glucose and inflammation in the brain. And we haven't heard anything, though, about insulin. And like our mom was in a trial, which was like an inhaled insulin, so that it went directly to the brain, because that was the place that they wanted to try and help.
So we actually funded that study, the first study, for Suzanne Kraft when she was at the University of Washington several years ago.
This is Yale.
And we funded that study. It was positive. It was several years ago. She did intranasal administration of insulin. The idea was, in part, that it could actually remove the amyloid because it inhibited an enzyme that degraded, that prolonged the degradation of amyloid. I'm not sure that makes sense, but we did fund the first clinical trial of intranasal insulin. She was able, from that repurposing small exploratory study, to get a very large NIH grant. She ran into trouble with the device. Yeah. So it's taking a long time to get that trial done. The other thing that we're doing is, because we think that intranasal insulin might be neuroprotective, we're just funding a clinical trial of intranasal insulin for people who have that postoperative cognitive decline. So after surgery, you get a little snort of insulin into your brain to protect you against the inflammation and the cognitive decline that happens after surgery in some people. So we've got a good history. We actually kind of created the field of intranasal insulin for Alzheimer's disease through our funding.
Thank you. Yeah, they did change the device, and this study was out of Yale. Our mom actually had the placebo, unfortunately, and the results were inconsistent, but it does sound promising.
Yeah, and I think you're raising a point about mom getting the placebo. A lot of patients come to us, and maybe somebody wants to comment on this, and part of the challenge is convincing people that it's worth it to go into a trial, because people often say, well, I'm going to spend 18 months on this drug, and I'm going to come in every month for an infusion, and I won't know if I'm getting a placebo. Why should I be in a trial? I don't know if you want to comment on anyone. I don't know if you want to comment on that issue. When you're recruiting patients, what do you tell them?
You know, I think it also brings up another point that just if we're going to solve this problem, we do need people to go into clinical trials and have a rigorous answer of whether something works or not. One thing we do know about participating in a clinical trial is that everyone gets better regardless of whether you're on placebo or medication. One thing that we try to do in our clinical trials is, if at all possible, offer people at the end of the trial who have been randomized to placebo the opportunity of using the open label medication. And that's something that can be put in some trials. And in other trials where it's a repurposing, they don't need any special way of getting the medication. So what we do is a third party can unblind the participant and let them know whether they're on medication, active medication, or placebo. And then we through our clinic, will help provide that person with the medication to give it to them open label. But it's so important that you participate in the placebo-controlled trial and that you have that answer. And we know that everyone benefits from the added medical attention and the psychosocial support and all those other factors that go into participating in a trial. Even if you're on placebo, you're going to have some benefit from it, and you can get an open label, especially in a repurposing trial like we're speaking to today.
It's a great point,
Yeah.
a really great point.
Yeah, let's just reiterate what Kristen said. We have to go together as a community to advance science. And I think I tell the patients, you are just scientists as I am here because we're partners of discovery. We can't advance without your help. And we're deeply grateful above all to all those patients and those families who have come forth to advance science. And I think, as Kristen pointed out, there are other potential benefits that even if they take the placebo, the engagement, being excited to be in a trial, to interact with your physicians and other people more often. There's some studies that show their benefits as well, even if you take the placebo.
And any other questions?
Hi, thank you so much for taking the question. And I wanted to acknowledge the work you've done, because I think I read on the website that you've put in about $65 million into about 600 programs, so I think that's
It's
just
actually
amazing.
over 130 million.
That's even better.
Yeah.
But I know that late-stage registrational studies are very, very expensive, in the tens of millions of dollars. And so I wanted to ask, how do you look at those studies? How do you partner those studies? Because some of the drugs like Riluzole, I think are generic, and so it might be difficult to get a commercial partner. So just wanted to know how you think about those late-stage studies.
Yeah, so the question, it's a great question. I'm glad you brought it up. The question is, like, we have positive trials here. Where's the commercial interest to actually bring these to market as an indication so that patients will have access? In other words, I could prescribe Riluzole or Rasagiline or Nabilone off-label in my practice today, and I have done some of that for patients. But what we really need is to somehow get this into a commercial mechanism so that a big company can let millions of people know about it and make it available. And that is a challenge. We were talking with Cures Within Reach people this morning about mechanisms. I mean, there are ways to do it through creating new intellectual property, through reformulation of the drug, through creating prodrugs, through getting data exclusivity, which means that if you have a positive trial and you go to the FDA for an indication of a particular drug for Alzheimer's, if it's a repurposed drug currently on the market, you get 3 years of what's called data, 3 to 5 years of what's called data exclusivity, which means that no one else can market the drug during this period of data exclusivity. It's not like 10 to 12 to 15 years that a pharma company would get for a new drug that came to market that was not repurposing, but it's good enough for a lot of investors to earn $200-300 million or whatever it is to do that. So we actually want to take on this challenge. We have 16 repurposing studies going on. We have positive results. Our next challenge is to work with the investigators and the university. We've been talking with the University of Toronto Technology Transfer Office. Should we create a biotech company that's dedicated to developing the indication of Nabilone? And another thing is, and I think this came out of the Riluzole study at Rockefeller, it stimulated Takeda and the tri-institutional program there to do new chemistry. So if we get a proof of concept that Riluzole, which is, as Anna said, is a glutamate drug, we know the mechanism, we know the biology, we can stimulate new chemistry, and new chemistry will lead to new drugs along that pathway. Anna?
Yeah, absolutely. I think the first thing is to have data and have those trials, to have data that confirm or test out if those drugs are truly effective. I think that's the first thing. And if they are, if you identify a novel mechanism, then it's very exciting, a novel target, and you can develop novel molecules that are much more efficacious in the future. So I think novel chemical entities that can have a more clear commercial path.
You know, the journal at Rockefeller, the journal that's put out by Rockefeller University is called the Journal of Experimental Medicine, and it was originally, as Anna mentioned, it was John D. Rockefeller actually established it as a place for human medical research. The first studies there were trying to develop better vaccines for human beings, and it led to the discovery of DNA. A long story, very interesting. But you know, what we're talking about here is human clinical research. We've cured mice with Alzheimer's like 600 times now, and none of that's really translated. Mice, I like to say, don't write books. You know, this, as Anna, a cognitive neurologist, has been telling you, I mean, the human mind is complex and unique, and we need to do these human experiments to find out if these drugs can work, and then that opens up a completely new area of research. And so this effort, and it's not a lot of money to do, I mean, relatively speaking, it's not like $400 million for a phase 3, but we can get these studies in these phase 2A, so to speak, clinical exploratory studies done for much less than that in practical terms, something that the ADDF can afford. Yes?
Good morning. I wondered, we read a lot about the influence of the gut on the brain, and I wondered if you were supporting any research in microbiome.
We're not. I think the microbiome is kind of interesting, but I'm not sure of its relationship to cognition at this point. There's a company called Cortexzyme out there that is testing a hypothesis around sort of gut gingivitis and so on. Maybe somebody would like to comment on the microbiome and Alzheimer's disease.
I just came from the American Neurological Association meeting a few weeks ago. I think there's the growing data that there's an important influence and relationship between the microbiome and cognition. I think it's early stages and it still have to identify which microorganism and do further testing in vivo, in animal models, and until we come to patients, as Howard pointed out, it's critical to human research, right? We want to cure patients. So, but I think the data is growing and is interesting, but it needs a lot more evidence.
Well, we have to go to lunch. I want to thank the panel. I want to thank everyone in the audience for joining us. It's been great.